Okay, let's dive right into this. Welcome to today's Explainer, where we're going to unpack this fascinating chapter of biology that literally transforms our entire understanding of life. We're moving from a simple mystery to a highly precise mechanism. Think of explaining the chapter as, well, kind of a detective story. We're going to retrace the steps of the early scientific pioneers as they moved from that basic mystery of heredity, like why do we actually look like our parents, all the way to uncovering the physical, biological mechanisms happening at the microscopic level inside our cells.
So here's our roadmap for this investigation. First, the mystery of heredity. 2, Mendel and his peas. 3, when the rules break. 4, the crossing over solution. 5, extra-chromosomal traits. And finally, 6, forward versus reverse genetics.
Section 1. The Mystery of Heredity.
Now, this fundamental mystery begins with something you've probably thought about sitting right there at your own dinner table. Seriously, think about this. Have you ever noticed that all the members of your family have several features in common? Why is it that you share certain facial features, or maybe hair color or skin color with your parents and siblings? For centuries, humanity just sort of observed these characteristics running in families. We knew there was a genetic basis, but the actual how and why, well, that remained a total enigma.
To solve this, we first need to define our core term, heredity. It's simply the transmission of characters from one generation to the next. What we know now, and what the brilliant scientists we're about to discuss actually helped prove, is that these inherited characters are physical instructions. They're present right there on our chromosomes in the form of genes.
Moving on to Section 2, Mendel and his peas.
So who was the first person to actually crack this code? Meet Gregor Johann Mendel. He's universally recognized today as the father of genetics. Born in Austria in 1822, Mendel started his monumental studies in 1843 while living as a monk. He was surrounded by lab facilities and just had this intense passion for research, so he set out to understand exactly how traits were passed down. And his model system of choice? The common pea plant, Pisum sativum.
And this brilliantly illustrates the sheer scale of his dedication. Mendel didn't just casually glance at these plants, right? He performed artificial cross-pollination experiments for 9 long years. 9 years. He carefully isolated and tracked 7 distinct pairs of contrasting traits, things like seed shape, flower color, stem height. By meticulously counting the offspring, generation after generation, he deduced the presence of invisible factors, which, of course, we now call genes, that predictably determine an organism's traits.
Section 3. When the rules break.
Now, Mendel had established these amazing laws of segregation and independent assortment. But, as you know, science is rarely perfectly neat, and researchers soon realized the rules were about to break. Mendel's rules basically suggested that traits sorted themselves completely independently of one another, but genes don't always play fair. In the early 1900s, shortly after Mendel's work was widely recognized, scientists like Bateson and Punnett started doing their own experiments, this time on sweet peas, and man, they threw a massive wrench into Mendel's established models.
See, Bateson and Punnett fully expected to see Mendel's classic 9:3:1 ratio when looking at 2 traits simultaneously, specifically flower color and pollen grain length. But look at the reality of what happened. They found that more than 50% of the offspring kept the exact same trait combinations as their parents. The genes for red flowers and long pollen were somehow sticking together rather than assorting independently. Basically, the math wasn't mathing.
Section 4. The crossing over solution.
This bizarre deviation absolutely baffled scientists at first. But the solution to the puzzle ended up revolutionizing biology entirely. The answer? A phenomenon called linkage. We understand now that genes aren't just floating around freely. They're anchored on chromosomes. And if the gene for flower color and the gene for pollen length happen to be located super close together on the exact same chromosome, well, they act like a package deal. They're physically linked, so they inherit together.
But wait a minute, if they're physically linked, how did Bateson and Punnett get any new trait combinations at all, even if it was less than 50%? Well, that happens because of crossing over. During cell division, specifically meiosis, homologous chromosomes actually pair up and exchange physical parts of themselves. It's like shuffling a genetic deck of cards, creating entirely new non-parental combinations that we call recombinants.
Now, what's really interesting about this is the literal physical proof that this happens. Classical experiments by scientists like Harriet Creighton and Barbara McClintock used genetic markers to observe chromosomes structurally swapping pieces. So it wasn't just some abstract mathematical theory anymore. You could actually see the chromosomes crossing over under a microscope. No way, right?
And scientists quickly realized they could use this crossing over rate as a very practical tool. They defined 1 centimorgan, or 1 map unit, as representing exactly a 1% chance of recombination between two traits. By measuring how often traits got separated during crossing over, scientists could literally draft the very first physical maps of chromosomes. It's pretty straightforward. The further apart two genes are, the more likely they are to cross over.
Section 5. Extra-Chromosomal and Sex-Linked Traits.
The discoveries just kept rolling in. Thomas Hunt Morgan provided massive validation for linkage and recombination using Drosophila melanostra, the humble fruit fly. When Morgan mated flies to look at body color and wing shape, he didn't get independent assortment either. Analyzing the offspring, he saw 83% of the flies kept the parental combinations, while exactly 17% showed new combinations due to crossing over. Morgan's work firmly proved that certain linked traits were even tied directly to the sex chromosomes of the fly.
And then came another massive plot twist in explaining the chapter: extra-chromosomal inheritance. For decades, everyone just assumed that all your genetic instructions were locked safely inside the nucleus of the cell. But actually, scratch that. It turns out DNA is also hiding out in other cellular structures, specifically the mitochondria and in plants the plastids. Because the egg cell provides all the cellular machinery during fertilization, any traits controlled by this DNA are inherited strictly from the mother.
A totally perfect example of this maternal inheritance is the 4 o'clock plant, Mirabilis jalapa. Its leaves can be green, white, or variegated. But here's the kicker. The color isn't determined by standard Mendelian genetics in the nucleus at all. It's dictated entirely by the genes inside the mother plant's plastid DNA. Whatever phenotype the female plant has for leaf color, the offspring are going to inherit exactly that.
Section 6. Forward versus reverse genetics.
So we've covered incredible ground today, from Mendel's monkish garden to the inner workings of maternal plastids, and the crucial point is understanding how our modern scientific approach has evolved because of all this. Take a look at this comparison. Mendel, Bateson, and Morgan practiced forward genetics. They observed a phenotypic variation, like a visible trait, say a white eye or a wrinkled pea, found the mutant allele and worked backward to locate the gene. But today, because we've mapped these chromosomes so thoroughly, we often use reverse genetics. We start with a specific DNA sequence or gene intentionally mutated and then observe what phenotypic variations pop up to figure out what that gene actually does. It's like solving the puzzle from the inside out.
Which leaves us with this final, crucial question. How will these foundational discoveries of heredity, linkage, and crossing over continue to shape our understanding of life today? Think about it. From Mendel's patient counting of peas to mapping real-time crossing over events, every broken rule only revealed a deeper, more beautiful mechanism hiding inside our cells. By understanding heredity, linkage, and recombination, we literally hold the keys to modern medicine, agriculture, and our very biological identity. Keep exploring, keep asking questions, and I'll catch you in the next explainer.